Every living cell, from the simplest bacterium to the neurons firing in your brain, has a guardian, a flexible border that separates life from chaos. This border is called the cell membrane, a living dynamic structure that decides what enters, what leaves, and how the cell communicates with the world. In this video, we'll break down the fluid mosaic model, understand the lipids, proteins, and carbohydrates that make it up, and see why the membrane isn't just a wall, but a smart living interface. From biology, we understand that a cell is the basic structural unit of life. Now, what is a cell membrane? The cell membrane, also called the plasma membrane, is a thin, flexible boundary that encloses the cell and maintains its internal environment. The modern understanding comes from the Fluid Mosaic Model, proposed by Singer and Nicolson in 1972. If you imagine a mosaic floor, your different tiles of shapes and colors forming one surface. Now make that floor fluid, where the tiles can move around like boats in water. That's your cell membrane. It's a phospholipid bilayer, two layers of lipid molecules with proteins and carbohydrates floating within it. Together they form a dynamic semi-permeable barrier. Please pause and study this diagram. Composition of the cell membrane. All membrane consists of a double layer of lipid molecules in which proteins are embedded. The membrane contains 42% lipids, 55% proteins, and 3% carbohydrates. Let's start with the lipids. Lipids' main lipid component is phospholipids. Each has a hydrophilic head, water-loving, and hydrophobic tails, water-fearing. In water, they automatically arrange into a bilayer. Hydrophilic heads face the water, hydrophobic tails hide inside. This arrangement creates a selectively permeable barrier. Small non-polar molecules like oxygen and carbon dioxide pass freely, but ions and large polar molecules like glucose or sodium cannot. They need help. Proteins in the membrane. Proteins, as shown in this image, make up more than half the membrane's mass, and they're the real workers here. They're of two types, lipoproteins and glycoproteins based on position and attachment. They are divided into integral and peripheral proteins. Integral proteins. These are embedded within the lipid bilayer, tightly held by hydrophobic interactions. Removing them usually destroys the membrane. There are two main types. Intrinsic monotopic proteins embedded in only one side of the bilayer, attach firmly but do not span the entire membrane. As you can observe here, these integral proteins do not span the entire bilayer. Transmembrane proteins. These span across the whole bilayer, from extracellular to cytoplasmic side. They have hydrophobic regions that interact with lipid tails and hydrophilic regions exposed to the watery environments on both sides. Important note, all transmembrane proteins are integral, but not all integral proteins are transmembrane. However, all intrinsic are integral proteins. Here in this image, you can see that the monotopic integral proteins, which are the intrinsic proteins, span just one side, while all others are transmembrane, spanning the entire layer. Peripheral proteins greater than. These sit loosely on the inner or outer surface of the membrane. They attach indirectly, usually via interactions with integral proteins or the polar heads of lipids. Now, because they're not deeply embedded, they can be easily removed without damaging the membrane. Here's the peripheral protein, and as explained earlier, attached to either polar heads or integral proteins. Let's look at some functions of membrane proteins. Intrinsic proteins often serve as enzymes for catalyzing reactions. Peripheral proteins form part of the cytoskeleton, maintaining the cell's shape and providing structural support. For instance, sodium-potassium pump is a transmembrane protein that maintains ion balance, carbohydrates in the membrane. Carbohydrates form only about 3% of the membrane but play vital roles. They're never free-floating, always attached to lipids, forming glycolipids, or proteins, forming glycoproteins. Together, these form the glycocalyx, a carbohydrate-rich coating that acts like the cell's ID card. It helps cells recognize each other, communicate, and even protect against mechanical and chemical damage. Cholesterol is a steroidal lipid, amphipathic, meaning that it has both hydrophilic and hydrophobic parts. Its main role, regulation. It prevents the membrane from becoming too rigid in the cold and too fluid in the heat. Think of cholesterol as the thermostat of the cell membrane, keeping it at the right balance of fluidity, stability, and permeability. Functions of the cell membrane. The cell membrane does far more than act as a barrier. Its major functions include 1. Protection, forms the cell's outer boundary and shields internal structures. 2. Digestion and excretion, helps take in nutrients and remove waste via vesicles. 3. Selective permeability, allows only certain molecules, especially non-polar ones, to cross freely. 4. Insulation, due to lipid content, it prevents electrical leakage in excitable cells. Organization, provides a framework for the arrangement of proteins in specific functional areas. 6. Cell junctions, links adjacent cells through junctional complexes to form tissues. Quick quiz recap. All right, let's test your understanding.
Feel free to pause the video.
You'll have 5 seconds to answer each Q1. What does fluid in the fluid mosaic model mean? Answer is B, the ability of membrane components to move freely within the layer. Q2, why do phospholipids form a bilayer in water? Answer is C, the hydrophilic heads face water while hydrophobic tails avoid it. Q3, what do carbohydrates do in the membrane? Answer is D, they act as identification and communication markers. Q4, which molecule passes most easily through the bilayer? Answer is C, oxygen gas, non-polar small molecule. Comment how many you got right. So the cell membrane isn't just a passive shell, and it's a living interface managing protection, communication, and control. Every heartbeat, every nerve impulse, every immune response starts at this microscopic frontier. If you found this helpful, don't forget to like, subscribe, and turn on notifications. Because next we'll explore how substances actually move across this membrane, diffusion, osmosis, and active